I said the last one was the last for the day. I'm gonna do one more. Then it's the last for the day. Yeah. This next one is from Ron. And how are you doing? Yeah. This is someone we know and like. All right. So, question, and it's a nice, succinct question. Thank you. How are data and samples collected to identify the presence of or prevalence of various COVID strains, in particular the Delta variant? So it's known that Delta is becoming the dominant strain in various places. How do we know that? Great question. Yeah. And from the outside, it would seem like people are making this decision of what variant you have based on symptoms or based on, like you can tell, oh, there's a person's green as a watermelon, they must have Delta, and that's definitely not the case. The symptoms are not a reliable guide to which variant that you have. Yeah, that was almost English, but I'm trying my best. Yeah. Let's see. So how it's actually done is by collecting samples, and the samples will often come from whatever sample you're giving and having tested for COVID. So the calling what variant is a thing that happens after a sample tests positive. It's a very expensive process, which is why we don't just try to variant call as a way of diagnosing whether you have the disease. Yeah. It would just cost, I don't know, 10 to 100 times as much to get the same answer with slightly more information. That what are you going to do? There's still no clinical application for knowing what strain of coronavirus a person has at the moment. You don't treat them differently if they have Delta versus Alpha, Beta. In terms of actual patient care, if it's the first case of a new variant in an area, there may be, people may be a little more on their toes about making sure it doesn't get out, or watching for secondary infection, spreading out. But yeah, there's nothing really actionable about knowing it's Delta versus not Delta. The samples come from a saliva sample, and then what has to happen is you take the saliva sample or nose swab sample, whatever. If it's a nose swab, you're going to dilute it in a little bit of salt water to get some of the nose sludge that has the viruses in it off there. And then from that point, you need to open up all the viruses that you've got and get out the thing inside, which is a long coiled piece of messenger RNA, because that's what a coronavirus is. It's a big twisted up messenger RNA inside of a little spiky ball. Yeah. And then once you have the messenger RNA out, and there are different ways to do that, you can then begin to break it into little fragments and read out each of the fragments. And there are, again, many ways to do this, many different technology platforms that are all really interesting. And I guess the result is that, in some of these, you have little pores, and you're basically sequencing little strings of the RNA that you turn into DNA by pulling it through a pore really fast and reading out. So the pore is going to change a little bit. It'll change charge and change shape potentially a little, depending on which nucleotide is going through it at the moment. And so you can tell it's got an ACGT, whatever, something like that, going through. There are others where you have little fatty droplets that have tiny styrofoam beads that have little branchy chains that pick up bunches of copies of the same thing, and then you can read that out. There are some that work with light, where you will get the sequencing to show a little burst of light every time it puts in a nucleotide, and so okay, I'm putting on the A nucleotide now, and you see little lights flashing here, and here. You can say, okay, each of those things I'm sequencing just added an A, so we'll make a note of that. And then, fine, wash that off. I'm going to put some C on there, and then some G and some T. And sorry, I just whacked the microphone with exuberant gesturing. So there are several processes. Generally, it's going to be a thing called next-gen sequencing, which is already kind of a last-gen term for what it is. It's just really fast sequencing of samples where you don't know what the sample is. And several competing ways to do this, all of which give you pretty good results, but each of which give you they're slightly better at one thing than another, or give you a slightly bigger piece, slightly smaller piece, but more of them. Yeah, whatever. They all work really well. And so that's how it happens. And then usually, so this is something we do. We will then take the sequence data and we will upload it to a database. And as you upload it, it runs it through a little checker and it compares it to all the different variants and everything that's out there. And so it'll say, okay, this one's delta, 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 delta. This one's alpha. This one is a mess. You didn't do a very good job at this one. And it'll give you quality control and tell you what each one is. And then at no point during that process does the person who is sequencing the virus and reading out the information, at no point do they actually know who the person was, or whether they were particularly sick, or whether they're better now, or whether they died. And the whole process usually takes about a month between you spit in the tube and it was Delta variant. And by that point, usually one way or another, the infection is over. So, yeah. It's slow, it's careful, it's science. That's where it comes from. And scientists are able to do this because it is essentially a useful public health surveillance technique. But in order to even do this on samples that were collected for a different purpose and are basically going to be thrown away if they're not sequenced, you still have to go through all kinds of approval processes. And I think there was a video I did maybe a week ago that was talking all about the sorts of committees you go through and what you need to satisfy them about. Because in sequencing patient material, there is definitely a layer of bioethics there. And you don't want to mess up the bioethics when you're trying to help, because that's not the goal, is it? So thanks very much. Very good question. And that's how the sausage is made. Yeah. And so based on that, we know that something like 90% of the sequences that were sampled in July and have been sequenced already are Delta. And this is worldwide based on all the sequences that are there. So these are the people that got their work in as fast as possible. Based on the number of sequences we got for July and what we had for June, and the increased number of cases that we see in July compared to the number worldwide that there were in June, I would anticipate that we have maybe 10% of the data so far in for June, and maybe it's even a little bit less than that. So the picture could change, but right now it is 90% Delta. And so, the only thing that's likely to happen is that you're likely to start getting more Delta plus variants, which are going to start from Delta, because that's the one thing that's out there, and the virus can only work with whatever genes and versions of those that it inherits. And then you'll see it making mutations that go beyond that. And some of those may be bad for us. Some of those may be good for the virus. I don't know that any of those will be good for us, or anything that's bad for the virus just gets wiped out so fast that you wouldn't be likely to see those persisting in the world, because the viruses are fighting against each other to some extent. It's a little bit indirect, but that is what happens. And a new variant can appear and completely wipe out old, well-established variants very quickly. Only takes a couple of months. Happened several times already with COVID, and it's happened several times already with different flu strains, that we know about. So, it's a dog-eat-dog world out there. And I say this as a dog owner. Dogs eat all kinds of stuff. Thanks very much. This has been Ask Dr. Ben.